This is our DNA. It's made up of two strands that contain a variety of letter sequences that serve as the blueprint for life. Unfortunately, there are various threats to our DNA that can create breaks and damage this blueprint. Fortunately, our cells have developed methods to combat these effects and repair the damage so that we don't lose precious information. For simplicity, let's depict the two strands as just two parallel lines. There are many sources both within our cells as well as external agents that can cause damage by creating a break in both strands. One method that our cells use to repair these breaks relies on homology, or in simpler terms, similarity within the DNA sequences. In this image, the regions indicated by the green boxes have the exact same letter sequences as each other. We'll call these repeat sequences. One mechanism that our cells employ when a break occurs between repeats is a process known as single-strand annealing. In this pathway, various proteins are recruited to the damaged site to repair the break. The first set of proteins work to chew back one strand on each side of the break. The newly exposed single-stranded DNA is now vulnerable, so another protein will attach itself to protect the DNA from further damage. The next protein's role is to bring the two repeats together, and because the sequences are identical, they can line up and bind each other perfectly. A protein complex then arrives to cut off the unnecessary DNA tails that have been generated. Finally, the last set of proteins work to fill the remaining gaps and seal the nicks, resulting in the fully repaired DNA product. One method scientists use to study these proteins and this repair process is fluorescence microscopy. In fluorescence microscopy, the DNA damaged site, as well as any proteins of interest, are labeled with different colored fluorescent markers. In this example, the damaged DNA has been labeled red, while two separate repair proteins have been labeled yellow and blue. This technique allows us to see and track the proteins as they make their way to the damaged DNA. Using a fluorescent microscope, we can see the different colors localized to the same location within the cell, indicating that the proteins are present at the DNA damage site. This technique is just one of many methods used to study the specific and intricate process our cells undergo to maintain the integrity of our genome.